Hello. In this presentation today, we shall be talking about meristems. What are meristems? The plant tissues are basically divided into two types, namely meristematic and permanent. Meristems are a type of tissues found in the growing regions of the plants. It comprises of undifferentiated mass of cells capable of active cell division. The term meristem was given by Nageli. Looking at the characteristic features of the meristems, meristems are small, they are young, immature, and divide rapidly. Plants continue to grow throughout their entire life cycle because of this unlimited division of meristematic cells. The divisions are mitotic in nature. The cells are living, small, oval, rounded, or polygonal in shape, with a prominent nucleus, and are compactly arranged without intercellular spaces between them. These meristematic cells have the ability to enlarge, stretch, and differentiate into other tissues and organs of the plant body. They are responsible for the wound healing processes of the plant. Meristems can be classified into different types based on their stages of development and origin, based on their location. Let's first look into the classification based on the stages of development and origin. Promeristems. These are the young regions of the plant body which lay foundations for new organs. Promeristems are also called primordial or embryonic meristems. These cells have thin wall, prominent nucleus, and dense cytoplasm. They could be vacuolated or non-vacuolated. Promeristems give rise to primary meristems. The primary meristems, as told earlier, are derived from the promeristems and constitute the primary permanent tissues of the plant body. The primary permanent tissues being the parenchyma cells, chlorenchyma cells, etc. They are found distributed in the shoot, stem, root tip, and leaf primordia. The primary meristems, after growing for a sufficient period of time, may lose their ability for cell division. Under certain conditions, they may regain their power of cell division and thus become secondary meristems. The secondary meristems are lateral in position and are responsible for giving rise to secondary tissues. One best example is cambium, which is a secondary meristem. Next, we have the classification of meristems based on their location in the plant body. They can be divided into apical meristems, lateral meristems, and intercalary meristems. The apical meristems are located at the shoot and root tips. That is the growing regions of the plant body. So they bring about the elongation of the roots and shoots. These give rise to the primary plant body. The lateral meristems are located at the side or lateral regions of the plant stem or root. They bring about an increase in the stem girth and thickness. These are also known as secondary meristems because they are responsible for secondary growth. Example, the vascular cambium and the cork cambium. The intercalary meristems are the detached portions of the apical meristems separated due to the growth of an organ. They occupy the internodes or the base of leaves. These meristems help in increasing the length of the internode and are basically seen in monocotyledonous plants. Classification of meristems based on their function in the plant body. This was put forth by Haberland. So primary meristems at the stem and root apices are segregated into three types, namely protoderm, procambium, and ground meristem. The protoderm is present towards the outside or outer region and is differentiated, or it differentiates into the so-called epidermal tissue system. The procambium develops into vascular tissues, namely xylem, phloem, and cambium, while the ground meristem gives rise to the hypodermis, cortex, endodermis, pericycle, and pith. So this is the image of the shoot apex, where you can see the young leaf primordia. This is the image of the root apex. Coming to the structural organization of the shoot apex. Three theories were put forth to explain the organization of the shoot apex, namely apical cell theory, histogen theory, and tunica corpus theory. The apical cell theory was proposed by Nageli in 1944. According to him, the apical meristem comprises of a single apical cell which is large, dome-shaped, and inverted. The apical cell is tetrahedral in shape and has 3 or 4 cutting phases. The side of the apical cell that is directed upward is triangular or squarish in shape. The cutting phases of apical cell divide by asymmetric division and form large packet of cells which differentiate and form different segments of shoot. The apical cell theory was not accepted since different parts of the plant body have independent origin. This theory was not applicable to gymnosperms and angiosperms. The histogen theory was proposed by Hanstein in the year 1868, according to which the apical meristems are divided into 3 zones of initials or histogens, each of the histogen giving rise to a particular tissue zone. The 3 histogens are dermatogen, periblem, and pleurome. Dermatogen is the outermost layer of the apical meristem, which gives rise to the epidermis of the root and stem. The periblem gives rise to the cortex of the root and shoot, as well as the inner tissues of leaves. The pleurome gives rise to the central cylinder of stem and root, including the pith. It is the central core of stem and root. This region is surrounded by dermatogen and periblem. To be noted, in the roots, the dermatogen also cuts off some cells called calyptra, which gives rise to the root cap. Tunica corpus theory, as indicated in the picture here. The theory was proposed by Schmidt in the year 1924, and this theory is concerned with planes of cell division in the apex. It is applicable only to the shoot apex and not to the root. According to this theory, the shoot apex comprises of 2 tissue zones, namely tunica and corpus. Tunica is the outer zone of shoot apex and is characterized in having anticlinal plane of cell division. That is, the division wall is laid down perpendicular to the surface, resulting in 1 or more peripheral layers. Dicotyledons exhibit 1 to 5 layers of tunica, while monocots have about less than 5 tunica layers. The corpus is the inner zone of the shoot apex and comprises of cells that are several layers deep. The initial cells of corpus occur below the tunica. They show both anticlinal as well as periclinal divisions, producing a mass of cells which grow in volume. It is a corpus zone which is destined to give rise to the cortex and vascular tissues. Thank you.